Alright, welcome back. This video is going to be on section 3.34 and 3.6 and is going to be about the lipids, carbohydrates, and nucleic acids. So, when we looked at last time carbon compounds, we're now able to split it down into four main pieces or main groups of biological molecules that make up all life. And today we're going to talk about carbohydrates and lipids and nucleic acids, and we'll talk about proteins next time because there's a lot more to say about those, and it kind of needs its own video. But I like to start out with this little concept map because it works pretty well at distilling kind of the pertinent differences between the three, so what they're made up of, and we'll definitely look at those today, and the elements that are made up of all of these three compounds as well. So if they have carbon or hydrogen or oxygen or some other things in them too. So this is a good thing for you to look back at, and I'll actually be giving you a sheet on Wednesday that has these but without the names in them for you to kind of fill in when you're working through them. So as you looked at 3.1 and 3.2 on Monday, you saw that the dehydration synthesis and the hydrolysis reactions were really important because that's how we join monomers of biological molecules into bigger pieces into polymers. And for all of the ones we talk about today, lipids, carbohydrates, and nucleic acids and proteins next time on Wednesday, this is how we build them and how we break them apart. So each of these little balls would represent a monomer, like a monosaccharide in carbohydrates, or a nucleotide and nucleic acid. On branching off of them, we have different functional groups, and some of them are hydroxide groups like this, and some are hydrogens. And so basically what a dehydration synthesis reaction is when the hydroxyl group of one little unlinked piece comes close to the hydrogen group of another unlinked piece. That hydrogen combines with the oxygen, or the hydroxide, and they both kind of break off and form water. And while they do that, they link the pieces of the shorter polymers together so we get a longer one. And that's why it's called a polymerization reaction. It makes a longer polymer from these smaller spots. And guess what? We can do the opposite of that too. We can break the longer polymer apart by adding water. So we add water to that long polymer, and the water breaks into hydroxyl and hydrogen groups, and one piece of the polymer that is getting broken in half right here grabs the hydroxyl group, and the other grabs the hydrogen, and then they get broken in two. So now you have two smaller polymers, and this is how we build and destroy all four of those classes of macromolecules. So it's kind of a big portion. Number one of today is seeing how these four types of molecules all utilize the same process to build and destroy bigger polymers. So the first one we're going to talk about is carbohydrates right here. And is remember kind of what those are. Those are sugars. So all of our examples are going to include sugar. So for example, these are very important because they're used as energy or fuel in living organisms, and that we can also use them to build structures, biologic structures. So like plants use them to build those rough, rigid parts to maintain their shape, and crustaceans use them to build their shells. And carbohydrates contain carbon, hydrogen, and oxygen. So these two pictures on the right are both carbohydrates. They're both the singular building blocks. Remember, all of these are polymers made up of building blocks, and the carbohydrate building block is a monomer, or monosaccharide. Saccharide means sugar. Easy way to remember that. And these monomers, these monosaccharides, can either be three 5 or 6 carbons long, 3 being triose, 5 being pentose, and 6 being hexose sugars. So this one right here, 1, 2, 3, 4, 5, 6 is a hexose sugar. And it is that building block of longer carbohydrate polymers. Now you'll notice these building blocks can come in two different forms. There's this long one, we call it a chain form, or this kind of hexagon shape, we call it the cyclic form. And actually the cyclic form is what happens, and it's a reversible reaction that's really complicated, we won't get into. But sometimes when you put certain chain forms in water, this will happen, and this is actually glucose right here. So we'll notice that we do have a carbon backbone, and then coming off of it we have hydrogen and hydroxyl groups. Same thing in the long chain form. And so carbohydrates, the monomers, all have these hydroxyl groups coming off of it. And you'll notice if you really have your functional groups down, that we have a carbonyl group right here on the chain form only. So that's another way to differentiate the chain form and the cyclic form. A really easy way to identify, if you're seeing it on the page or on the test, and it's like, is this a lipid, a carbohydrate, or a nucleic acid? You can count up the carbons, you can count up the hydrogens, and you can count up the oxygens. And all of the carbohydrates, if they're monomers or polymers, will usually have for every one carbon, they'll have 2 hydrogens and 1 oxygen. This 1 to 2 to 1, 2 to 1 ratio. So that is a very easy way to remember or identify a carbohydrate. 1 carbon to 2 hydrogen to 1 oxygen ratio. So we can build longer carbohydrate chains from our monosaccharides. We can join them together to make two disaccharides. They have 2 sugars tied to them. And those, ta-da, coming back, are formed or put together by our dehydration synthesis reaction of our 2 monosaccharides. So for example, this picture on the bottom, we have 1 glucose molecule plus another glucose molecule, and right here, right where it has in blue, that is the hydroxyl group of 1 and the hydrogen of the other that are going to be pulled off and form water. And then this oxygen right here that's left over is going to form the bridge or the link between the two right here. And this in carbohydrates is called a glycosidic linkage. That oxygen hanging out in there in the middle. And so we get water from that dehydration synthesis reaction of 2 monosaccharides forming a disaccharide. So for this example, we got 2 glucose molecules forming glycosidic linkage, a disaccharide called cellulobiose, which is 2 beta glucoses linked together. And we can keep going, and we can keep adding different monomers or disaccharides on there till we get long chains of polysaccharides, or many sugars together. And polysaccharides contain many monosaccharides, so between 100 and 1,000, hundreds and thousands of monosaccharides make up a polysaccharide. And some examples of polysaccharides are starch, which is over here, starch, and glycogen. Starch is used for storage of nutrients in plants, and glycogen is what animals and humans use to store energy in our livers. So starch and glycogen look kind of like branch structures of glucose right here, store energy, and they can be used as building blocks, like cellulose in plants. Right here we've got some cellulose, and chitin, which makes the shells of like shrimp, and chitin is right there. You can see there are glycosidic linkages, that O hanging out like we're holding hands between the 2 monosaccharides right there are glycosidic linkages. And you can see that those glycosidic linkages can take place between the one, if we number the carbons going clockwise, the 1 carbon and the 1, 2, 3, 4 carbon, or it can be between the 1 and the 5 carbon. So it can be a 1, 4 glycosidic linkage, or 1, 2, 3, 4, 5, 6, 1, 6 glycosidic linkage. That's kind of beyond a lot what we're going to talk about with the AP test, but if you're being like, why don't I might see that in my book? That's why. So, lipids are the second, and they're not a polymer. Why is that? Well, they're composed, a lipid is composed of 3 different pieces, but you can't really put, once you have one whole, you can't really put them together. So, lipids kind of all share the fact that they're non-polar, and they're hydrophobic. So if they're not polar, they don't mix with water very well, they're hydrophobic. And we use them in organisms, biological organisms, to store energy. They have 9 calories of energy per gram more than any other macromolecule. And they can also provide protection. So we use them to protect our vital organs. They're around our vital organs, and they are used to make our cell membranes and to protect our neurons in our head. We make lipids through, coming back again, a dehydration synthesis reaction of 3 fatty acids and 1 glycol. So if you look at this picture right over here, this part right here is the glycol. It's 3 carbons with 3 hydroxyl groups, 1 on each carbon, and a fatty acid. This is a fatty acid right here. It's basically a hydrocarbon, so see a carbon bound to a carbon bound to a carbon, on and on and on, with hydrogens coming off of them, except on one end we have a carboxylic acid group. And what happens is the hydroxyl part of that carboxylic acid and a hydrogen for the hydroxyl group on the glycol or glycerol forms water, and then we get a link between both of them, like you see in this picture. We have an ester linkage, and another functional group is called an ester. We don't really need that for this class, but you can see that we make these fats by linking 3 fatty acids to our glycerol, 1 to each of those hydroxyl groups via dehydration synthesis. So in fats, we can kind of have 2 main types. We have saturated and unsaturated. So saturated fats, that fatty acid chain has all carbon-carbon single bonds, meaning they're saturated with hydrogens. No carbon can be bound to more hydrogen. It's a very long and straight, compact fatty acid. Unsaturated fatty acids have some double bonds thrown in there, so there could possibly be more hydrogens added. And it kind of gets this bent shape, and that's why they say you should eat more unsaturated fats, or if you're going to eat fat, it should be of unsaturated kind, because that bent shape makes it harder for them to stack up and stick together, especially in your veins, causing blockages. These saturated fats, they're straight, they can pile up and stick to each other, and they can block arteries and cause strokes and embolism, or and thrombosis and all sorts of stuff like that. But that's what's meant by saturated and non-saturated fats. Kind of the second form of a lipid are phospholipids, and they're really important because phospholipids make up the bilayer, the cell membrane. So these are really important because they're around all of your cells. And they're made up of 2 fatty acids connected to a glycerol via dehydration synthesis, just like this part right here. But the 3rd hydroxyl group of that 3rd carbon in the glycerol is connected to a phosphate group. So now we're adding a polar part, the phosphate group, onto our previously non-polar fatty fat. And so what happens is this head part, where the phosphate group is, likes water. It will mix with water, and so it sticks out towards the outside and the inside of the cell, and these nonpolar tails kind of get sandwiched together. So your membrane of all your cells is a phospholipid bilayer, so the heads of two phospholipids facing away from each other with the tails facing the middle part. In this way, these nonpolar tails can be as far away from the intercellular and extracellular fluids as they can. And the last form of lipids are steroids. So all a steroid is a lipid that fits this pattern of 4 fused rings, 4 hexagons and 1 pentagon ring. And they do a lot of things in your body, and one really important one is cholesterol, which is part of the cell membrane. So they're all made up of 4 fused rings with different functional groups attached to them. So here you can see testosterone, cortisol, and vitamin D and cholesterol all have these 4 rings that are fused to each other, but then they have different side chains and functional groups that go off of them, which change their shape and therefore their function and also their chemistry, the things that they can react with. And the last macromolecule we're going to talk about today is nucleic acids. So the nucleic acid is really important because they're involved with genetic information, the most important one being DNA and other being RNA. So DNA makes up your genetic code. It tells your cells what proteins to produce and when. And your RNA does a variety of things. RNAs can act as enzymes, but they also relay that genetic information from DNA and also help to translate and transcribe them into proteins. So the important thing to note here is that all nucleic acids are built of monomers called nucleotides. And these nucleotides are composed of one or two sugars. DNA has a deoxyribose sugar, so they have a carbohydrate in them. RNA is composed of ribose sugar, so it has a carbohydrate as well. And here's a comparison. The only thing that's different between those is the 2' carbon on the ribose has a hydroxyl group that is alpha, but the deoxyribose just has an alpha hydroxyl or a hydrogen and no hydroxyl group. Then all nucleotides have a phosphate group and one of 5 bases. So DNA has these 4, cytosine, thymine, adenine, and guanine, and these are their shapes, and RNA has uracil instead of thymine. So RNA has uracil and no thymine. And what you can notice about these nitrogenous bases, very important nitrogenous bases, they have nitrogen. So DNA has nitrogen in it. That's going to be important. Nitrogen is very important. So just like we'll talk about proteins, they have nitrogen in them. So what I say, you can notice that the nitrogenous bases come in 2 forms. There's ones that are single rings called the pyrimidines, and there's 2 that are called the purines. They have double rings. Looking over, probably one multiple choice question in every 3 years asks you to identify them. So it's probably something that's good to be familiar with, but not something you should kill yourself memorizing right away. So the nucleotide polymers in DNA and RNA, like we said, there's 1 phosphate, 1 of 2 sugars, and 1 of 5 bases that form a nucleotide. So a nucleotide is composed of this sugar, and off of the 1 carbon there's 1 of those 5 nitrogenous bases. In DNA it's adenine, thymine, cytosine, and guanine. In RNA you replace the thymine with the uracil, and off of the 5' carbon there's the phosphate group. It's always the same. 1' is always the base, 5' always the phosphate, and they're stacked on top of each other. So the 3' hydroxyl group and the hydroxyl group on the phosphate attached to the previous 5' carbon go through a dehydration or condensation, dehydration synthesis reaction. Remove the water and bind together. So the 3' end, 3 carbon end is always bound to the phosphate of the one below it. That's why we say that DNA is 5' to 3', or one end is a 3' end, The other end has the phosphate is the 5' end. And this goes for DNA and RNA. They're both polymerized the same way. The nucleotide right here, this is one nucleotide. A nucleotide is one phosphate, one base, one sugar. So what's in this box right here? Alright, now the structure. So DNA is double-stranded, like you'll see over in this picture over here. It's got two strands that kind of are in that, winding staircase-like pattern. It's called a double helix. It kind of winds around. And there's 11 bases, so 11 rungs every 365 or 360 degree turn. And the bases of one strand pair up via hydrogen bond, that's what these dots are, with the bases of the second strand. And they pair in the same predictable way. Adenine always pairs with thymine, A with T, and cytosine always pairs with guanine, C with G. And you can notice A and T only form 2 hydrogen bonds, and C and G can form 3. So the C and G linkage is the stronger one. You'll notice looking at this picture that the 3' end, so let's look at this deoxyribose sugar, 1, 2, 3' end is down here, and the phosphate 5' end, 5' is up here. Well, on the other strand, this is the 3' end and this is the 5' end. And that's what it's called by running anti-parallel. DNA always runs anti-parallel. The 5' end on one strand is opposite of the 5' end on the other. Now, RNA is pretty much the same. It's pretty much the same, and we'll kind of move this picture right here. And RNA is the same, except if we had an RNA picture right here, this half wouldn't be there. It's only single-stranded, and any place we'd have a T, like right here, we would have a U. So uracil and not thymine is in RNA, and that well, I'll let you figure out why that is, because we're actually going to have a case study that you're going to read about that. But go over this again when we get to our inheritance structure of DNA, but that is our DNA and RNA structure. So we'll go back to the beginning here. Let's remember the 3 things we talked about: carbohydrates, lipids, nucleic acids. They're monomers, nucleotides, fats, glycerol, and fatty acids, and monosaccharides, and then their composition. And on Wednesday, we're going to do some modeling so you can be able to get this solid for next time. Alright, so if you have any questions, just give me an email. But otherwise, I'll see you on Wednesday.